guys, so this video is going to be the beginning of chapter 1, just the very first section. Let's get started on biotechnology. So what biotechnology means is that we are going to use science to modify living systems to benefit people, to benefit humankind. And PCR is a type of biotech that we're going to talk about in the next section, but not this one yet. So let's talk about using microbes, the little guys, bacteria and viruses to genetically modify, genetically engineer other things. Before we do that, of course, case study. So we have Kayla. She is 23 years old and she's an electrical engineer and she likes to run. She moved from Arizona to New Hampshire and she loves to go for long runs in the forest. In July, she went hiking in the mountains, sounds like me. And in early August, she developed a low fever, headache, mild muscle aches, and fatigue. She took some ibuprofen, told herself she needed to sleep, get some more rest. We'll check back in with Kayla and see what's going on with her. All right, so genetic engineering. What are we talking about when we say genetic engineering? When we engineer something, we can talk about biotechnology in general when this comes to selectively reproducing what we want. Like our dogs, for example, we choose the genes and the breeds that we like because of the traits that they carry. That's biotechnology, that's engineering. That's genetically modifying an organism. Agriculture, that we have been breeding plants that we like because we like their offspring forever. That's genetic engineering. But what we're talking about specifically in modern day biotechnology is genetic engineering using recombinant DNA. So what we're going to do is we are going to take a host. So here we've got a bacterial host's genome in the form of its plasmid. So we talk about the chromosomal DNA, and then we have those small circular plasmids off to the side. So here's a bacterial's normal plasmid, and there's a gene that we want to use, and we want the product of this gene. And so in this case, it is the insulin gene. And if you did Bio 1010 or if you did Bio 1107 with us at Georgia Highlands, you probably did the plasmid project. And that's exactly what this is. So we need human insulin gene because before we had human insulin gene to produce, we had to get insulin from pig pancreases. And when we slaughtered the pig, we would harvest their pancreas and grind it up. And that's the insulin that we would use. And not everybody could handle porcine insulin. We needed human insulin. So now we can get that. We can have bacteria produce that insulin, produce that protein from human genes made for us, but they're done by bacteria. So we take that insulin gene from humans' chromosomes, carve it out with a restriction enzyme that cuts specifically in a certain place marked on the DNA, and it will cut there and only there. And whenever it sees that place, it's going to cut it. That's what restriction enzymes do. They digest and they cut them. Why do we have things that cut DNA? Because we might be invaded by viruses, and those viruses may be trying to hijack our cells. So that's part of our defense, is that if we see foreign DNA come in, we can use those restriction enzymes and cut it to bits. And then we're not susceptible. So we have lots of restriction enzymes. Bacteria have lots of restriction enzymes, and we can use a restriction enzyme to cut the gene around insulin. And then we will take that and have it incorporated. And as long as that restriction enzyme cuts here and cuts here, and it cuts the plasmid at one location, we can incorporate that in. And so we'll incorporate it in, and it is now called a recombinant because it is not the original host species DNA. It now has DNA from another organism. So it's a recombinant DNA. Specifically, if this is human and bacterial, this is transgenic. Transgenic, different species from the host. So this is a transgenic recombinant DNA, and we've got the blue plasmid with the red human gene in there. And we're going to put that into the environment. We could use transduction, we could use conjugation. There's all sorts of different ways that we can get that bacteria infected with this plasmid, but we are going to infect the bacteria. It's going to incorporate this plasmid in somehow. Multiple ways that we're going to talk about. Once it's got that plasmid in that cell, we can now use that cell, and every time it goes through its genetic processing and goes from DNA to RNA to protein, this cell is going to produce insulin gene for us and the insulin protein for us. which is what we want. So now all the recombinant bacteria gain the ability to secrete human insulin, and then we can just harvest that up. And it's a much better, easier, faster, cheaper way to do this. So this is genetic engineering. The example is using bacteria to synthetically produce their own human insulin. So when we talk about those restriction enzymes, I was telling you, like, they cut in very specific locations. They code for a certain sequence, and where they see that sequence, they are going to cut it. How do they cut it? They may use a sticky end, or they may cut it in a blunt end, but they will make a cut. Sticky ends are going to cut beep, 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 like that, and blunt ends are just like it says, straight across. So they can cut either one. Sticky ends, it would allow it for multiple hydrogen bonds across the way, so those guys can re-anneal and come back together. For this one, for the blunt ends, we would have to use, remember we talked about ligase as the DNA glue that glues the strands together. So we would use ligase to put these two blunt ends back together, rejoin the DNA all the way across. So we would have DNA over here, and we would connect it with ligase and DNA over here, and connect that with ligase. And now we've sealed back up our cut, assuming that we've put in our plasmid and we want to stick it back together. But these are restriction enzymes. They have sticky ends and blunt ends, and they will cut a specific sequence. Every restriction enzyme cuts a different place, a different sequence. How we can get things into the cells. We can use the plasmids as vectors, and there are some artificially constructed plasmids like these that have been constructed in the lab, this plasmid vector PUC19. And this guy, he is a polylinker site. He is going to have multiple places where it could get cut by restriction enzymes. So we can see, here's one, here's one. There are lots. EcoRe, the BAM, all of these are different restriction enzymes that will cut in different places. You can follow their colors to all the different places where they will cut along this restriction, this plasmid, so you can put lots of different things into this artificially constructed plasmid that will be incorporated into a bacteria later. Another thing that this plasmid vector has is going to be a reporter gene. So in this case, it is the LAC-Z, what is that, alpha? LAC-Z alpha gene. And with that LAC-Z gene is going to break down and metabolize lactose. So this LAC-Z gene, if lactose is present, this bacteria will be able to metabolize it. It can also, with the LAC-Z alpha gene, it can metabolize a synthetic analog to this X-gal, this fake chemical that it can metabolize. And when it does, if it metabolizes this colorless material, it will then change color. It will change color to blue. If it doesn't metabolize, then it's just going to stay clear. It'll stay white. But if it can metabolize, which it can because it has this reporter gene, it will not only metabolize the lactose, it'll metabolize this synthetic product, lactose, that it's going to break down. And when it does break it down, it changes color. So it's a way to report or show you this is working or it's not working. So we're going to see this process in the blue-white screening method that's going to be in the slide coming up. But then this is a synthetic plasmid that we can use as a vector. It's got multiple cloning sites, so it's a polylinker. MCS is the same term, different term for the same thing, meaning that lots of restriction enzymes are involved. They can cut in lots of different places, which means you can put lots of different things in this vector and use it to change up your bacteria's genetic information. Okay, so here's our molecular cloning, similar to what we did in our plasmid project for earlier biologies if you took them with us. So we had our bacterial plasmid, and so here's our genetic plasmid, and we said we had some foreign DNA. For us, it was the insulin gene, and so we came along and we cut our insulin gene using a restriction enzyme, and you needed to know for our project which restriction enzyme is the right one. Assuming you got the right one, you cut down the gene out of your strand of human DNA just to get the insulin gene. You've cut out the gene and you've got these sticky ends, and you need to also use that same restriction enzyme to cut your plasmid. So you're going to cut your plasmid. When you do, you're going to cut this gene. You're going to cut through the LAC-Z gene. So if you're cutting through the LAC-Z gene, is it going to work if you cut a gene in half? No, no. So if you were able to get this plasmid to incorporate this foreign DNA into itself, you've now destroyed the LAC-Z gene. It's not going to work. So it's not going to change the color like we said. If it works, it changes that colorless molecule into a colored blue color. But if you destroy the gene, you cut it in half, it's not going to change the color. All right, so let's keep going. What else do we have in this plasmid? We have the LAC-Z gene that we just destroyed when we made it into a recombinant. But we also have this. Let's not forget this because this is important too. We have the ampicillin resistance gene. So in the ampicillin resistance gene, ampicillin is an antibiotic. So it has an ability to withstand the presence of ampicillin, and it can survive in a media that has this antibiotic that will kill off everything else. So this superplasmid can withstand ampicillin, and it can metabolize lactose. But now we just destroyed the lactose gene because we put it in a plasmid. And all of these things are going to matter when we put it through the test, through the screening. So we have created in the next step, we put the insulin gene in, and we've sealed it all together with ligase, and we glued it all together, and it's ready to go. And we now have these recombinant plasmids. So these plasmids are getting incorporated into these bacteria cells. I'm going to say they're E. coli. I don't know what they are, but I'm just going to make it up and say they are E. coli. And so we had the E. coli growing in the medium, and we're going to put these plasmids all in the medium. And there were different forms of horizontal gene transfer that the bacteria can incorporate new material, right? So one of those was transformation. So if you put the bacteria out and around the material, the DNA material out and around the bacteria, it's going to absorb it into its cell membrane and bring it in and be like, oh, this is cool stuff, I'm going to keep it. So that's what's happening here. This is transformation, and the bacteria take in our plasmid, which is what we want. We want them to take in the plasmid. So they can take it in with transformation. Some bacteria, I think it said Bacillus is naturally competent. It will do this automatically. It sees stuff in the environment. It's like, yeah, gimme, gimme. And then there are other ones out there that have to be artificially competent. So either there's chemicals or some sort of electrical impulse that causes their cell membrane to be more susceptible to bringing in the material before, because they weren't naturally going to bring stuff in. So they could be naturally competent, or we can artificially modify these cells to bring in the material. So anyway, the cells have brought in the material. That's called transformation. We talked about this before. They brought in the material with transformation, and now we are going to test and see, did it work? Because we're growing bacteria on this plate, but we need to know, is this the bacteria that has the plasmid, and is the plasmid working like we want it to? So we are going to put it on this media, and on the media, it says it is cultured on a plate, with ampicillin and a substance that changes color, like we said, that beta-galactoside enzyme. So if it's cultured on a plate with ampicillin, our bacteria that have that ampicillin gene are resistant to the antibiotic drug. If other things were trying to grow on this plate, they wouldn't, because the antibiotics would kill them off. So now we only have the bacteria that we've been culturing in the first place growing on here, because they're all resistant to ampicillin. So do these bacteria actually have the plasmid that we want them to have, or are they the ones without the incorporated gene? Do they not have the insulin gene, and they just have the regular plasmid without the insulin gene? We need to know, did they get the insulin gene or not? because that was the whole point of this experiment, was to see that these bacteria are going to incorporate the insulin gene and be able to produce insulin for us. So did they do that here? So the ampicillin eliminated all the other bacteria. We are down to just, I'm making it up, E. coli. We're down to just the E. coli. Now, did the plasmid take, is the insulin gene in there? We said if the insulin gene is in there, that it destroyed this LAC-Z gene because we cut it right in half right here. And since we cut it in half, it's not going to metabolize that LAC-Z, that colorless molecule, and turn it into blue. So all of the ones that are white and not blue, white colonies, have the recombinant plasmid because they couldn't metabolize that colorless substance and turn it into blue. These are colonies that do have the original plasmid that has the LAC-Z gene intact, and they are able to turn the LAC-Z into this blue color from colorless. So these do not have the recombinants. So we are growing E. coli on this plate, and only E. coli on this plate, but only some of them, the white ones, actually have the recombinant with the human insulin gene in them. And those are the ones that you're going to want to choose and grow more and more of. So we figured out a way to screen this and make sure that this molecular cloning was working. And this is through the blue-white screening method. And this bacteria was able to absorb these genes using transformation. There are other ways to induce genetic material into a new cell, and that is with conjugation, where we are going to use those fertility plasmids, or there's also transduction. And remember, transduction requires a virus, a bacteriophage. So somehow, some way, we can put this stuff in the environment and utilize transformation outside of the cell, and the cell will naturally bring it in. We can use conjugation, which was basically bacterial sex, where the other cell lassos it, creates a bond of cytoplasm between the two cells, and that plasmid comes across, that F plasmid can come across with conjugation. Or we can use transduction and use a bacteriophage, modify the genome inside the bacteriophage, which is called a phagemid, once we put in that recombinant material in there. And the phagemid inside the bacteriophage will inject that genome, that modified genome, into the cell and infect it that way. So 3 different ways to get recombinant DNA inside of a prokaryote. Okay, what if you wanted to keep track of all the genes that an organism has? That sounds pretty ambitious, but it's doable. And we have model organisms that we do have that information. We know all of the genes that this organism has. So what can we do? We can keep a genomic library. So we can take those genetic chunks, those chunks of DNA, and we can break them down into the pieces. And so now we've got DNA fragments, and we can take those fragments, and just like we saw before, we can make them into recombinant plasmids. And now each plasmid here and here and here has a different chunk of that genome. And then we're going to infect the cells, and we only grow, say, this genome, say whatever this organism is, some kind of worm, whatever this guy is. Maybe we wanted to know the gene that allows this thing to grow. And we determined that the gene that allows this thing to glow this super cool color is the green gene. It's the green one. And so now we can use just these out of the bacterial library, out of the genomic library, and just play with those bacteria. And we can tweak around and mess with the DNA that controls that gene and see if maybe we can modify it somehow and use it to our own purposes. So we would create a genomic library, all the genes in an organism, we bust them up into small pieces, and then each piece will go into different bacteria that we can play around with. We can also use this method to create a genomic library utilizing phages, utilizing bacteriophages. So we've got, here's our genome that we want to keep track of. Over here, we're going to incorporated it into the viral genome, into the phage genome. And now the phages, we're just going to say this is A and this is B and this is C, are going to infect the cells, and when they infect the cells, they take over the cells, right? And then they're going to go through their cycle where they are going to rupture the cell. And when they do rupture the cell, they're going to create a plaque. And now we can see all of the different plaques from this lawn here. And every plaque is a different color for the different chunks of DNA that were put inside of these phages. A benefit of using the phages versus the bacterial plasmids is that the particles that we can put in the phages are larger. We can put a bigger chunk of that library inside of a phage than we could inside of the plasmids. Another thing that we can do is look at, okay, so the genome is everything, right? The genome is going to include the genes that are coding for everything possible, the non-coding stuff, everything in there. What if we only want to look at the genes that are actually being expressed, the ones that the cell actually needs to use and express and send out there? So if we want to do that, we can, if we were looking at our cells and what our cells are expressing, we'd look at the mRNA. That's what the genome is expressing. We're going to go from DNA to messenger RNA, and then from the RNA, and we'll use that to translate into a protein. So we would look at our RNA, but we can't do that. So we will take that RNA and we will backtrack it. So we're going to use the mRNA and then expose it to the reverse transcriptase that goes backwards. Instead of going DNA to RNA, reverse transcriptase takes that RNA, backtracks it to DNA, makes the complementary DNA for that. So now from our reverse transcriptase, we are going to build complementary DNA. We will incorporate those into plasmids. And now we have complementary DNA for this structure, for this group of plasmids, for this group of bacteria, and for this group of bacteria. So these are the complementary DNA based off of the genes that are actually being expressed out of an organism. What if we want to mess with eukaryotes instead of just prokaryotes? Prokaryotes are relatively easier. They take the modifications easier than the eukaryotes do, but we can, and we are, and we have already been modifying eukaryotic DNA. And so when we do that, it's called transfection. So we have genetically engineered, I'm sure we all have heard about GMOs. So we have genetically engineered plants. We have the flavor saver tomato. There is Monsanto soybeans and corn, and all of that stuff is genetically engineered. So we are doing it, we have been doing it, and plants are eukaryotes. We have been modifying animals as well. The textbook mentioned pigs with increased nutritional value, which I have not heard of, but it also talked about goats that secrete products in their milk that we were looking for. I can't remember exactly what that product was that was released in their milk, but it was something in the goat milk that gets released that we have modified the genetic material to make the goats do so. So we can infect plants and we can mess with animals too. But when we do that, it's a little bit trickier. So one of the ways to do that is called electroporation. So we have our plasma membrane, just like the bacteria had the plasma membrane, and we said that some bacteria were, where's my slide for it? Some of these guys were naturally competent. Where did it go? Here we are. Some of these guys were naturally competent in that they would automatically incorporate stuff in there, and others were not. It took chemicals or electrical charges for their cell membranes to be modified to bring the stuff in. That's pretty much what we have to do for animals. That's what's happening here in this situation. So with electroporation, it is going to make pores, holes in the cell membrane of that eukaryotic cell. We're going to create holes here with this electrical shock. And with the electrical shock, we are also going to have a positive and a negative side. So inside the cell, we are going to make it positive inside the cell. So we made it positive and positive. And the information that we have, this DNA that we want to get into the cell, DNA is naturally negatively charged because of those phosphates on the end. DNA has nucleotides and it's phosphate, sugar, nitrogenous base. Phosphates have a negative charge. DNA has an overall negative charge. So if we put a negative charge on the outside, negatives and negatives repel. So it's not going to be outside the cell. It's going to be attracted to the positive charge inside the cell, and it's going to zoom on through that pore in the cell membrane and go into the cell. So that's one way that we can get that genetic information from outside the cell into a eukaryotic cell that normally would be like, no, stay out. So this is electroporation, one method for us to get information into the cell. We can manually inject it with a micro glass pipette. So you can put that bad boy in there. Sometimes you will see this image when we think about, this makes me think about anyway, artificial insemination, where you have the sperm and it is being, the nucleus is being injected into the egg cell. That is micro injection with a glass pipette. Other things, this one always gets the chuckle in the face-to-face class because it's a gun. It's a gun. It makes me laugh. So it's shooting beams of stuff at cells. So with plant cells, compared to animal cells, plant cells have a cell wall. So their cell wall is another barrier for that information to have to get through to get inside the cell. So they will dissolve the cell wall with some enzymes, and that is going to create a plant cell called a protoplast because it's removed its cell wall. So we remove the cell wall, it's this little protoplast, and they use this gene gun that is going to shoot gold, where is it, gold or tungsten particles, and then they coat the gold or they coat the tungsten in the DNA that we want to be inside this cell. So we blast, we shoot, we beam gold particles into the cell. This just cracks me up that you can do this, really. So the gene gun is another way to get modified genetic material inside of plant cells. TI plasmids, these are ones that are used in our transgenic plants. So TI is coming from Agrobacterium tumefaciens. And this bacteria, when it infects plants, it normally naturally infects plants. And when it does, it is going to cause tumors in plants. That's why it's got tumefaciens in its species name. So what they do is they take this plasmid from the bacteria. They're going to take out the gene that makes the tumors, but they're still going to keep that plasmid, and they're going to put in the gene that they want in its place. So we have our TI plasmid. We're going to put our gene of interest, and we're going to put it in here. And now we have destroyed the gene that makes the tumors because we cut it in half when we stuck in this good gene that we wanted, whatever the good gene is that we want. And now we have this recombinant that is going to go back into, we're going to get A tumefaciens to incorporate this plasmid back into its cell. And then we're going to take the agrobacter and we're going to infect the plant with it, with this modified recombinant plasmid. And when we do that, that gives us a recombinant plant. So this plant is infected with the bacteria, and the bacteria's genome was modified. So now it becomes part of the plant's cell's genome. Right here, using a TI plasmid. So we are doing this. I want to say this is what I think Monsanto's TI corn is, but you may have to Google it. I may be wrong, but I want to say their corn is using this kind of method for their genetic engineering. There's another one that I left off, viral vectors. We're going to get into that in the last section of this chapter, where we use viruses and mess with their phages, and we put our human good genes in there, and those human good genes, whatever that gene is that's messed up in the person who is ill, we're going to take a healthy gene and use the virus to infect the cells and incorporate the good gene in the hopes that the good gene will take the place of the damaged, miscoded gene in the person who is ill. And this is gene therapy. We'll talk more about it at the last section of this chapter.